Welcome and thank you for joining the 2021 Houston Area Parkinson's Society Summer Lecture Series August session. I'm Kathleen Christ, Executive Director of HAPS. Since 1974, HAPS has been the only organization in the area providing comprehensive services to the local Parkinson's community. Our mission remains the same since we started, to ensure that no one faces a Parkinson's diagnosis alone. Today, we wrap up our HAPS Summer Lecture Series with our August session. For the past 11 years, HAPS has hosted this fun, summer-themed educational event. It gives us the opportunity to hear the most up-to-date Parkinson's information from experts in our community. Due to ongoing considerations related to COVID-19, we decided to keep our educational programs virtual this year, and we are looking forward to resuming in-person educational events soon. Before we get started, I have several groups to thank. First, I want to thank the individuals who make up our board of advisors, and medical advisory board, as well as the members of the HAPS team for their ongoing support and the role they play in developing and implementing our programs. The combined effort and energy of these groups has allowed HAPS to maintain and expand our reach and services over the years. Next, I want to sincerely thank our sponsors. We are so fortunate to have so many wonderful partners in the work that we do. This group has remained steadfast in their commitment to supporting the local Parkinson's group and to helping us ensure continuation of our mission. Specifically, I want to thank our presenting partner, Supernus, as well as AbbVie, Accorda Therapeutics, Boston Scientific, UCB, Amneal, Home Instead Senior Care, Medtronic, Synovian, and Acadia. Additionally, I want to thank all of you at home for your wholehearted participation in today's program and in all that we do here at HAPS. You are the most important part of the work that we do. And finally, I want to thank our presenters for sharing their time and expertise today, helping us gain a better understanding of the disease and for getting us up and moving after today's lecture. All right, so quickly, I want to give you a rundown of the day. You're going to hear from our speaker, Dr. Joshua Shulman, who will then join me for a question and answer session after his presentation is finished. But before we join Jeffrey for his exciting movement lab. If you do not already have the handouts, you can access them on the HAPS website at hapsonline.org under the Summer Lecture Series section, or by clicking the link sent in the email that contained the link to today's webinar. That's the email that came from HAPS, not the one that came from Zoom. The program includes the full presenter bios, upcoming HAPS events and programs, information from our sponsors, and the presentation for today's lecture. We're going to take a couple of brief breaks while we move to the question and answer portion of the program, and then again before we go to the movement lab with Jeffrey. So I thank you in advance for your patience with this whole process. During the question and answer portion, we'll be addressing the questions that the audience, that you all at home, submit through the throughout the course of the lecture. So if you're participating in today's live program, then you'll be able to put all your questions in through the little Q&A button at the bottom of your screen. We've had a little bit of some technical problems this morning. Dr. Shulman is off-site and is joining us from his office, and we've had some difficulty getting him logged in. But so you're going to hear him say things like next slide, because I'll be sharing my screen and advancing his presentation for you. So just bear with us as we work through some of this. But at this time, I'd like to introduce you to Dr. Shulman, who is an associate professor of neurology and neuroscience and molecular and human genetics at Baylor College of Medicine, investigator in the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital, and director of the Baylor Center for Alzheimer's and Neurodegenerative Diseases. He received his bachelor's degree in biochemical sciences from Harvard College and his PhD in genetics from Cambridge University. He studied at Harvard Medical School and MIT Division of Health and Science Technology, where he received his medical degree. Dr. Shulman completed his neurology residency and fellowship training in the Harvard Partners Program at Brigham and Women's Hospital and Massachusetts General Hospital in Boston. Dr. Shulman's research focuses on understanding the genetics and pathogenesis of neurodegenerative neurodegenerative disorders, including Parkinson's disease and Alzheimer's disease. In addition to being widely published and serving on a number of scientific committees, he remains clinically active caring for patients with Parkinson's and Alzheimer's and related disorders. So welcome, Dr. Shulman.
Thank you so much, Kathleen. I hope everyone can hear me okay, despite our technical difficulties. It's a great pleasure to be able to present to HAPS again today. And no, you are not hallucinating. This is, in fact, a lay. Of course, all neurologists wear lays like this, as you know. So I think Kathleen is going to bring up the slide deck. Perfect. And I'm going to jump into a lecture to you all on Parkinson's and the promise of genetics. And, when I've done this in the past, we've been very interactive. We've had some of my assistants moving around the audience with microphones. Obviously, that's not going to be possible today, but I do think on the positive side, I understand we're able to reach probably a much larger audience via Zoom. So I'm super excited to teach all of you a little bit about Parkinson's and the promise of genetics. So Kathleen, please advance the slides. So wanted to give you a quick overview of what I'll talk about. I think this is a pretty unique lecture. You're going to get a very high level understanding, I hope, of genetics and Parkinson's disease and how it may affect you. I am going to start by talking just a little bit of Genetics 101 about what genes are, how genes vary within the population. Then we'll talk about what the heritability of Parkinson's disease, what we know about how the genes affect the risk for Parkinson's. And I'll try to make some distinction between thinking about genes as causes versus risk factors. We'll also talk a little bit about how genetic factors might modify Parkinson's disease. And we'll end with I think, some useful and practical information about how currently genetics figures into clinical care for Parkinson's disease, how you can obtain genetic testing, why you might be interested in that. And then we'll end with some case studies that represent many of the patients that I've encountered and the types of testing results they've uncovered. And I hope to do this all in about 40 minutes or so. So next slide, please. So I need to start by just teaching you a little bit of biology. You've probably heard of genes. Genes are made up of DNA, and DNA is really like the recipe for all of our cells and for everything that makes us human. Each of our cells has 23 pairs or 46 chromosomes. So chromosomes are all the genes that are wrapped up into this larger structure. This figure is showing you what a chromosome looks like. And if we zoom in on it, ultimately we get to a strand of DNA, which you may have heard is like a letter code, a recipe book that's composed of C's, A's, T's, and G's. There are about 30,000 genes in the human genome, which is what we call the collection of all the genes stored in our chromosomes. And these genes are the recipes for what make the proteins in all of our cells, including the neuron cells that make up our brain. Next slide. These proteins really are what drive all the functions of the neurons in the brains in our brain. So it's ultimately what allows us to do everything we do, from thinking to moving. So in thinking about the genetic code, which is in our DNA, as I mentioned, and as you may have heard, it's very much an alphabet code composed of these 4 letters, A, T, C, and G. And so in this schematic cartoon, you can imagine that there is a code within the genes that is the normal sequence. And then over time, for a variety of factors, changes in this code can occur. When a single of these letters changes, so in this example, a T that's in red changes to C, we call this a point mutation. And so you've probably heard the term mutation. Mutation is really the substrate of everything that makes all of us different from one another. So mutations aren't necessarily good or bad. They are the substrate for all of the variation in the population that is biological in nature. But of course, changes like this or mutations can also sometimes affect risk of disease or even cause disease on their own. A single change like this in the massive genome is sometimes sufficient to cause disease, including Parkinson's disease. Next slide, please. So I mentioned that the genes are packaged into chromosomes. And what you see here is an actual photo preparation of all the human chromosomes. And so they're little strings packed up, strings of genes, and chromosomes are paired in human biology. So there are 23 pairs. The 23rd pair are the X and Y chromosomes, which are the sex chromosomes. Those are shown at the bottom right. And one thing I wanted to show you about these human chromosomes is, besides the kind of point mutation that I was referring to on the earlier slide, another kind of important genetic change, including one that can affect risk of Parkinson's disease, we call structural variation. So whereas a mutation that is a point mutation is a change in that letter code sequence that makes up the genes, we also can have larger deletions or duplications of regions of the chromosome. And so you can identify those type of changes often on a photograph like this by just looking at the chromosomes and realizing that one is longer or shorter than it should be. And so a cartoon is shown for this at the right here, where you can see a normal chromosome and then an example of what a duplication might look like, where a patch of genetic material, which could include one or many genes, is actually duplicated. These types of changes can happen in the normal course of reproduction and biology. So a duplication like this or a deletion could also affect risk of disease. And we see that these types of changes can affect risk of Parkinson's disease. Next slide, please. So, if you go back just a couple decades, people thought that genes had nothing to do with Parkinson's disease. Well, we now understand that, and this came from this observation that most patients with Parkinson's disease, about half of patients with Parkinson's disease have no known family history. I would say about 15% of cases we consider familial, meaning that generation after generation, we can identify relatives who are also affected with Parkinson's disease. But still, the majority of cases are sporadic. And it was for this reason, in part, that we thought genetics probably doesn't play a dominant role, because it was only a minority of cases where generation after generation, the disease would turn up repeatedly. However, fast forward to 2021, the case is really quite the opposite. We now believe that virtually all cases of Parkinson's disease are genetically influenced. And I choose that word carefully because I suspect in most of you, it's not that Parkinson's disease has been seen generation after generation, but many of you probably have some relative, perhaps a parent, an uncle, a cousin that is also affected by Parkinson's disease. So we appreciate that there is an influence that comes from the genome on Parkinson's disease risk. And even in those cases where you have no known family history, we now feel that it's likely that genetics plays a role in almost everybody. And I can explain more how that might work. Next slide. So in classical genetic thinking about risk and the way genes pass on, we recognize two main forms of inheritance. And inheritance simply refers to the way genes are passed on from one generation to the other. These two forms of inheritance are known as dominant or recessive inheritance, and they're shown in this cartoon here. And so imagine on the left that you have two parents, a mother and a father, and that the father is the carrier of a risk gene, such as a gene that causes Parkinson's disease. So in this cartoon, if somebody is colored in purple, that indicates that they have Parkinson's disease. And you can see that the father here is a carrier of a Parkinson's disease mutation and actually manifests Parkinson's disease. And 50% of this individual's children will be at risk of also developing Parkinson's disease because there are two versions of the gene. I mentioned that chromosomes are paired. And so in this example, there are two versions of the particular gene that we're talking about. One has a mutation that causes Parkinson's disease. And so there is a 50% chance that that particular mutated version will be passed along. That's shown in the schematic. On the left, we call this dominant inheritance. On the right, there's an alternative example in which if you have a single mutation, you do not develop Parkinson's disease, but you need to inherit a copy of that mutation, that abnormally functioning gene from both parents. And so in the case of a recessive inheritance model, and that's what's being shown here, it's only 25% of the progeny or the children coming from a pair, if both are carriers, that actually will have both copies. And that's what's shown in the boy that's represented on the right side. That's the first of the four children that are had. That individual will be predicted to develop the disease like Parkinson's if the gene is inherited in a recessive fashion. And you can see that two other children out of the four are only carriers of the mutation. And so in a recessive model, those individuals would not develop Parkinson's. So the upshot is that Parkinson's disease is a genetically complex disorder, and there are examples of genes that obey both of these rules, both the dominant form of inheritance and the recessive form of inheritance. And I give you examples here. So you may have heard of the alpha-synuclein gene, or you may have heard of the LRRK2 gene, LRRK2 gene, if you've read about Parkinson's disease genetics before. Those are examples of dominantly inherited genes. And there are also some examples of recessively inherited Parkinson's causing genes. One such example is called Parkin, and I'm showing that here. You have all of these slides in a handout, so if something is unclear, that handout can be accessible to you. You can review it later. We'll also, of course, have a question and answer period. So next slide. I want to talk a little bit further about specific example genes. One important gene is alpha-synuclein, I just mentioned. Mutations in alpha-synuclein can cause dominantly inherited Parkinson's disease. And I'm showing you here on the left what we call a pedigree. A pedigree shows a specific family. So this is actually 5 generations of a pretty famous family in Parkinson's genetics. A circle represents a female, a square represents a male. And you can see that in the second generation from the top, the filled in circles represents where Parkinson's appeared. And you can see that in every generation subsequent to that, there were many individuals who had Parkinson's disease. And in that last generation, you can see that there are 7 individuals who actually had Parkinson's. These mutations are very rare cause of Parkinson's disease, but they cause this dominant inheritance pattern. And in the lower left, this is an example of DNA fingerprinting, which you might have heard of, which allows scientists to determine whether the gene mutation is present or not. It turns out that the alpha-synuclein gene, besides mutations, can also be duplicated. That's a kind of structural variation that I referred to earlier. And I'm showing you an example of that in the picture shown in the bottom in the middle, where there are 2 dots that represent the normal 2 copies of the alpha-synuclein gene. But then there's a 3rd dot towards the top, a red dot on a blue chromosome. That indicates a duplicated copy that shouldn't be there. And that can cause Parkinson's disease as well, that will pass along in families in a dominant fashion. So alpha-synuclein is very important as a gene because the protein that this gene codes for forms the defining pathology of Parkinson's disease, which is called a Lewy body, which you might have heard of. The Lewy body is an aggregation of too much alpha-synuclein protein that accumulates in the brain cells, particularly dopamine-containing brain cells. And I'm showing you an example of that in the bottom right. So this is a rare cause of Parkinson's disease, but alpha-synuclein protein is important in everybody who has Parkinson's disease because this pathology is really how we define the disease. Next slide. Another gene that I want to give you an example of is LRRK2. This is another dominantly inherited gene. And so in the upper right, I'm showing you an example of a family that has a LRRK2, LRRK2 mutation that causes Parkinson's disease. And so one thing that's interesting here that I want to highlight is that in the third generation, okay, you can see that individuals 1, 3, and 5 are filled in. They have Parkinson's disease. You can see that there are 4 children here, 1, 2, 3, 4 in generation 3. But you can see that neither of the parents actually developed Parkinson's disease. However, the father's mother or the maternal grandmother of these children in generation 3 did have Parkinson's disease. So this is an example of a pedigree where it looks like Parkinson's disease skipped a generation. Okay, because we can intuit that the father here in generation 2, which is labeled with a 1, and I put an asterisk there, that individual should be a carrier of LRRK2, of this mutation that's being passed along, but he somehow escapes Parkinson's disease. So this is a well-known genetic concept. We call this incomplete penetrance. It's when somebody carries a gene mutation that should cause disease, but it doesn't manifest, and it leads to a skipped generation like this. And in complex diseases like Parkinson's disease, unfortunately, incomplete penetrance is really the rule rather than the exception. And so LRRK2 is an example of a dominantly inherited Parkinson's disease gene, but it is incompletely penetrant. And for that reason, not everybody who has LRRK2 Parkinson's disease will have a well-established family history. It can sometimes cause sporadic disease. Another concept that I wanted to emphasize on this slide is that the penetrance of the mutation for LRRK2 that causes Parkinson's disease actually varies by ethnicity, and that's what's shown in the lower right here. If you happen to have a LRRK2 mutation and you're of Norwegian descent, which is being shown at the orange line, the penetrance is lower, and people tend to get the disease later in life at older ages. But in those who are of Tunisian or Ashkenazi Jewish descent who carry LRRK2 mutations, the curve shifts to the left and penetrance is higher. So this is age-dependent and ethnicity-dependent variable penetrance due to LRRK2 mutations causing Parkinson's disease. Next slide. So you might ask, and I want to take a pause for a moment, why should I care about Parkinson's genetics at all? And so this is a bit of an interlude where I'd like to try to convince you that genetics is important. One reason is that genetics tells us a lot about the mechanisms of Parkinson's disease. And as an example of that, I want to just highlight that most of what we know from basic research in Parkinson's disease comes from animal models like mice and even fruit flies. And we could not make these types of animal models unless we had a genetic handle on the disease. So for example, on the left here, I'm showing you a mouse that has a manipulation of this alpha-synuclein gene. And when you manipulate that gene in the mouse, on one side of the brain, you can see that the dopamine-containing neurons that are important in human Parkinson's disease also are lost in this mouse model. And so you can see that at both lower power, kind of a zoomed out view, and also higher power when you zoom in, the cellular density is lower. And that's because the dopamine neurons are lost. Interestingly, when you manipulate the alpha-synuclein gene in a fruit fly model, believe it or not, we do have fruit fly models also of Parkinson's disease, and some of my research uses these models. And we look at dopamine neurons in the brains of a fruit fly. When we manipulate the alpha-synuclein gene, we lose those cells as well. And that's what I'm showing you here underneath the fly. Also, I don't know, Kathleen, if you can click, there's a video that I'm showing you there at the far right. If you're able to just click that, what you'll see is that our alpha-synuclein flies actually don't climb as well. They're slower and they take longer to climb up a vial compared to our healthy control flies. So animal models have been hugely informative to understand disease mechanism, even when we're studying genes that only rarely affect individuals in the broader population of patients with Parkinson's. Next slide, please. Now, of course, understanding mechanisms also, we always hope, brings us closer to therapies. And I think Parkinson's disease is a fantastic example, especially in the last few years, of how genetics can help us identify and accelerate the development of drugs, therapies that can be applied towards the disease. And so in the case of Parkinson's, there are now therapies and trials that are underway for alpha-synuclein, LRRK2, and another gene called glucocerebrosidase, or GBA. And here I'm just highlighting one example of a therapy. This is still in a very, very early phase clinical trials where antibodies are being given to subjects enrolling in these trials. And these are antibodies that bind the alpha-synuclein protein. And I referred to alpha-synuclein protein earlier as aggregating to form this pathology called the Lewy body. And so the idea of this therapy is that these antibodies might be able to disassemble the Lewy body or prevent its spread within the brain and hopefully stop the progression of Parkinson's disease. Again, this is very early stage clinical trial work, but it's really remarkable when you think that when I started medical school, Parkinson's wasn't even known to be a genetic disorder. It was thought all to be due to things in the environment. And so here we are, less than 20 years later, and we're actually developing drugs to some of these genes that have been discovered. Next slide. Okay, so I mentioned this in my outline overview, but I'd like to just try to make a distinction for you all between thinking of genes as causes or risk factors. And so, when in Parkinson's disease, we clearly have examples like alpha-synuclein of rare mutations with very high penetrance that clearly will cause disease. They're sufficient. If you inherit that dominant alpha-synuclein mutation, you are almost certainly going to develop Parkinson's disease. However, on the other hand of the spectrum, there are also many more common genetic variants that have very low penetrance. And we really can think of these type of genetic factors as population risk factors, because if you have them, you have just a more modestly increased risk, but on their own, they're not certain to cause the development of Parkinson's disease. And one example of a gene like that is glucocerebrosidase, or GBA. You might have heard about it or read about it in relation to Parkinson's genetics. If you have a mutation in the GBA gene, you have a 5 to 10-fold increased risk of Parkinson's. Now, that may sound like a lot, and it is a significantly increased risk, but keep in mind that that increased risk is compared to the background population risk. And what that comes to is that only about 10% of all the carriers who have GBA mutations develop Parkinson's disease by the age of 65. And that's what's shown here on the lower right, where we're looking at carriers of GBA mutations over time. And you can see, even as they approach the healthy age of 100, it's only the line is only getting up to about 20% cumulative risk of developing Parkinson's disease. So it's important to keep this in mind when thinking about what it means to carry a Parkinson's disease risk gene. There are now literally more than 100 known genetic risk factors for Parkinson's disease. And most of them actually cause a much more modest risk than even GBA. GBA is a particularly common and strong risk allele. But I'm trying to highlight here that if one has such an allele, an allele is another word for a variation or a mutation. Even if one has this GBA mutation, there's certainly not a certainty of developing Parkinson's disease. So next slide, please. So we have now more than 100 Parkinson's disease genes. And this is just a way of summarizing that we can think of the frequency of those mutations or variations in the population. I'm showing that on the X horizontal axis of this plot. And then we can think on the y-axis or the vertical axis of the plot, what is the penetrance of that particular variation or mutation? And with a plot like this, it just allows you to think about the complexity of all the genes that cause Parkinson's disease. We have a few genes that if you have them, they're nearly sufficient to develop Parkinson's disease. Those are the high penetrance causal gene mutations. And then we have a much larger number of low penetrance risk factor types of gene mutations that are much more common in the population. But if you have them, your overall increased risk is quite low. And most people who develop Parkinson's disease with these types of genes probably have more than one. They act in combination. You can think of these types of risk factors in some way, like the way that you might think of what the risk of lung cancer is if you smoke. Hopefully no one smokes, but you might have thought about, you're aware that if you smoke, you have an increased risk of lung cancer. It's not certain that you're going to develop lung cancer if you smoke, but there is an increased risk. I think a lot of these genes can be thought of in a similar way. Next slide. And so, if you want to put this all together and you think about the spectrum of Parkinson's disease from familial to sporadic disease, those that pass along through families due to the genetic factors, and those in which they just crop up and there may not be a family history, we can kind of think of a spectrum of different gene mutations, and I'm showing some examples here along the horizontal, different mutations, but different mutations are associated with different amounts of risk, and that's the penetrance, and it's indicated in blue here. And so some mutations at the far left of this diagram have a very high amount of risk associated with them. They're high penetrance, and so you probably need very little else to develop Parkinson's disease. But at the other end of the spectrum, there are variations and mutations that are associated with very low risk. They're incompletely penetrant, and we have to think about the fact that other factors, including environmental factors, perhaps other unknown genetic factors, interact with those genes to push people towards developing Parkinson's disease. And we can't forget the important role that age plays, which is probably the single most important risk factor for developing Parkinson's. Next slide. Okay. So I want to just introduce one more complexity, and that is that although we tend to think of Parkinson's as one disease, and we tend to lump everyone together and say, you have Parkinson's disease, what we really appreciate, I can tell you I appreciate from being a neurologist that takes care of almost exclusively Parkinson's disease patients, is that really no two patients are quite alike. And that's because Parkinson's is highly heterogeneous. And this slide is just illustrating some of the ways that Parkinson's can differ from one person to the next: age of onset, the speed of progression, how much tremor or gait symptoms people have, the risk of falls, non-motor symptoms like developing cognitive impairment, how well people respond to medication, whether levodopa-induced dyskinesia develops, and how much disability accumulates over time. We see incredible variability amongst all of these factors. And it's becoming increasingly apparent that genetic variation probably plays an important role in some of this variability as well. Next slide. And so here I'm just giving you two examples of that. On the left, there are now studies that establish that the GBA mutation, the glucocerebrosidase gene mutation that we talked about earlier, not only affects the risk of developing Parkinson's disease, but in those who have the GBA form of Parkinson's disease, they tend to progress modestly faster. So the illness seems to have a more rapid internal clock, and that's what's shown on the left. And on the right, I'm showing you a study that looked at dozens of more modest, incompletely penetrant risk factors that are common in the population. And they looked at individuals who have more than one of these factors to accumulate an increased risk burden. And the study is complex, but it's showing that the more of these common risk factors that you have, there was a measurable uptick in the pace of progression of disease. So Parkinson's disease genetic factors also may modify the disease course, the speed of progression, and some of this heterogeneity. Next slide. Okay, so this is the part of the talk that many of you probably are going to be most interested in. So if you were dozing off, now's the time to wake up, because this is where I want to talk about some of the practical aspects of how Parkinson's genetics might be relevant personally for patients with Parkinson's disease. So until really quite recently, genetic testing was really very rarely considered in the diagnosis or management of Parkinson's disease. And I have to admit, I've been taking care of patients with Parkinson's disease for 10 to 15 years now. And it's very, very rare that I've ever had reason to send a genetic test in caring for one of my patients until quite recently. And that's because really we reserve genetic testing for quite unusual circumstances, such as cases where individuals develop Parkinson's at a very young age, like a childhood onset case, or in situations like the alpha-synuclein gene family that I mentioned, where people had very strong family histories. And we had very strong reason to expect a strong genetic effect. But this is rapidly changing. And as a geneticist who cares for patients with Parkinson's disease, I have to say we're at a real transition point. And I think the main driver of this, one, is our increase in genetic knowledge about Parkinson's disease. And two, we now have clinical trials that are developing and beginning to recruit for some of these genetic forms of Parkinson's disease. And I referred to these earlier, that some of these gene discoveries are leading us to think about new types of therapies. And to really start some of these therapies, we need to understand what the genetic causes of Parkinson's disease are in the people we recruit into our trials. And I just indicate on this slide, you'll have this in your handout, some resources to find some of these clinical trials. You may have heard of the Michael J. Fox Foundation's Fox Trial Finder. You can search for that on Google and find it quickly. And also clinicaltrials.gov. These are resources to identify where clinical trials are that might be recruiting individuals with specific genetic forms of Parkinson's disease. This is now absolutely the case for both the Parkinson's disease caused by glucocerebrosidase or GBA mutations, as well as individuals who have Parkinson's disease caused by LRRK2 gene mutations. Next slide. So many of you may have heard about direct-to-consumer genetic testing. So an example of this is the company 23andMe. And there are many companies, both 23andMe and others, that will offer the opportunity of genetic testing. You don't even need a doctor. You can go onto a website, send a saliva sample, pay a particular price, and get some genetic test results. My message to all of you is that I don't recommend these direct-to-consumer genetic tests. I don't think they're going to harm you, but you may be wasting your money. Some of my concerns are that most of those tests that are offered are quite incomplete, so that it's hard to interpret a negative result because many genes and mutations might not be tested for. Also, there have been some independent, rigorous analyses of results from some of these direct-to-consumer tests, and they actually have a very high error rate, including false positives. So, I like to say caveat emptor, which means buyer beware. A lot of these companies don't use the same rigorous quality assurance standards that we expect from clinical tests. And so I certainly have had patients who come to me with results from 23andMe and similar companies, but it can often be very hard to know how to advise patients based on those results. Those results will not be sufficient for the type of clinical trials that I mentioned earlier. And then what ends up happening is that there may be additional costs associated with confirmation. And those costs usually, unfortunately, end up being passed along to you all or the patient, because insurance companies won't pay for them. Next slide. So the type of genetic testing that I do recommend, I would call clinical genetic testing. And so what I mean by this is that these are tests ordered by your neurologist. And one important benefit of this is that it allows you to have a conversation with your neurologist about the appropriateness of testing. Obviously, it allows your neurologist to help you interpret the results of the test. This is a rapidly expanding area. There are a number of different types of tests. I kind of cover some of them here. I do want to say that in most cases, these types of genetic tests in Parkinson's disease are not yet covered by insurance. Hopefully that'll change in the coming years, but these are usually costs that have to be paid out of pocket by patients. So I actually did want to give you some information about what these tests cost. There are two main types of tests. One are called gene panel tests. These are tests that look at specific genes or specific gene mutations. And the devil is in the details here in terms of what specific genes, how many genes, and what specific mutations are looked for. Those gene panel tests vary between $200 or $1,000. I'm trying to give you a ballpark here. Now, one interesting thing is that there are some now sponsored quote unquote tests. And so the company Invitae is currently offering a gene panel test for Parkinson's disease for free. And free, it really has a footnote associated with it. And I encourage you to, with your neurologist, review some of the fine print if this is something you're interested in. So basically, the Invitae company is covering the cost of this test, but they actually want some of your data for research purposes. So they are getting something out of it. But we have had patients who are able to get tested and it doesn't cost them anything. And Invitae does do a high quality genetic test. So, this is a test that I can potentially recommend. There are other companies that will do this type of gene panel testing, including Fulgent. Baylor Genetics is developing a panel. So there are many sources. I think Invitae is the one I'm aware of that is doing the testing for free right now. A different kind of test is called sequencing, or you may hear whole exome sequencing, which refers to the portion of the genome that's being sequenced. This is a much more comprehensive test. It looks at literally every single gene in the genome. So you can imagine this test is more expensive. It tends to vary between $1,000 and $5,000. The important thing with these exome sequencing tests is not just the actual performance of the test, but the interpretation of the test. And there are many companies out there offering this test. Now, for any genetic testing, I think genetic counseling is something to very seriously consider. And genetic counseling is something to consider even before you proceed with a test, but certainly for interpretation of the test. But I think having genetic counseling, if you're seriously considered or on the fence about whether genetic testing is appropriate, is a good idea because it can help you understand what the implications of the possible results that you receive will be. I'll just mention genetic counseling, unfortunately, is usually not covered by insurance as well. So that could be additional out-of-pocket costs. One company that I just want to mention, which is a partnership with Baylor College of Medicine, is Consultagene. They offer genetic counseling, including for Parkinson's disease genetics. It is an out-of-pocket cost, but counseling can be done even over Zoom or a teleconference type call. Now, you should be aware, if you haven't heard of it, of a research study called PD Generation. This is a study that is being funded by the Parkinson's Foundation. It's currently offered at Baylor College of Medicine and it's recruiting. You don't have to be a patient at Baylor College of Medicine to enroll in the PD Generation study. This particular study is offering clinical genetic testing for 7 different genes. It's actually using a gene panel assay performed by the company Fulgent. And genetic counseling is included in this study. So individuals speak with a genetic counselor after they receive their results, and it's explained what the implications of those results are. So this is a fantastic opportunity right now. And there are other sites. So if you look up the Parkinson's Foundation PD Generation's website, you can learn where this study can be accessed. And there are other research projects that might offer the opportunity to have high quality clinical genetic testing and return the results. We're developing a study at Baylor College of Medicine right now to do the sequencing test and return a complete and comprehensive result with genetic counseling to participants. Next slide. So I indicated that as a geneticist and a neurologist taking care of Parkinson's disease, it's really quite an exciting time. And I hope that some of you in the audience also feel excited about the opportunities with rapid advances in genetics. I think in just a few years, we're going to know all of the genetics of Parkinson's disease. I think things are moving so fast that we will know every last gene and how it contributes to this disorder. I think this is going to allow us to have much better models of disease, including better mice and better flies and other ways of studying the disease. And I think understanding genetics comprehensively is going to allow us to much better stratify the patient population. And this is going to lead to better clinical trials and hopefully improved and many new therapies. And ultimately, I think what we can expect from genetics, and I think this represents the real promise, is a much more personalized kind of care for Parkinson's disease. Right now, I think we generally tend to lump people together. There's a fairly formula way of treating people and advising them about their risk over the course of disease. But if we can understand more about their genetics, their risks, and how genes influence and modify disease, we can offer more personalized ways of treating them. Next slide. So this is a slide that just summarizes the main take-home messages. Genetics likely influences almost all cases of Parkinson's disease. In a minority of cases, a gene change may be sufficient to cause the disease, but more commonly, there's really a more modest relative increased risk from a gene variation. And we can think of these gene variations also as modifying disease progression or some of the specific symptoms that each patient develops. Genes help us understand disease mechanisms and hopefully pinpoint drug targets. And that's absolutely the case, we think, in Parkinson's disease. And genetic testing is changing very rapidly in the clinic setting for Parkinson's disease. And I think it's going to play an even more important role in the near future. So next slide, Christine. Sorry, Kathleen. So I'm going to end with, and I think I can do these pretty quickly because I see we're coming up to the top of the hour. We just start a little bit of late. I want to give you just a couple of example cases because they tend to tie together some of what we've talked about. So as we go through these cases, I want you to think a little bit about what you would do in each of these circumstances, and maybe you can kind of give me some feedback during the question and answer period about whether you would have want to obtain genetic testing in each circumstance. So next slide, please. So my first case, there are 4 of these, is Sue. She's 70 years old. She's diagnosed with Parkinson's after noticing a little difficulty getting dressed, some slowed walking, and some occasional stumbles. She realizes that her younger sister also has a mild tremor. And for this reason, Sue is worried about the risk of Parkinson's in her family. And so she chose to get genetic testing. Next slide. So in Sue's case, the results were the discovery of a LRRK2 gene mutation. And I'm showing you the specific mutation here. This is one of the causal mutations for Parkinson's disease. So just to kind of amplify that, if you have this specific LRRK2 mutation, you're quite likely, but not certain, to develop Parkinson's disease. And the estimated penetrance is about 85% by the age of 70. Now, interestingly, if you have LRRK2 Parkinson's disease, there's some evidence of reduced non-motor complication, like less risk of developing cognitive impairment. And but there's an increased risk of more gait difficulty and falls. So that gives you a sense of some of the personalized prediction that might be possible if we know about LRRK2 Parkinson's disease. Now, because this is a dominant mutation, Sue would be counseled that her children or her siblings have a 50% chance of sharing the LRRK2 variant, and then they would have that risk of that 85% possible risk of developing Parkinson's disease late in life. Next slide. So case 2 is Bob. He's 54 years old. He's diagnosed with Parkinson's disease after having some tremor in his right hand, and he noticed some changes in his walking with shuffling. Bob also mentions that his memory is not what it used to be. And he also brings to our attention that sometimes I see little people on the kitchen floor, but I know they aren't real. So obviously Bob is experiencing some mild hallucinations or Parkinson's disease psychosis. And Bob does have a family history His uncle developed Parkinson's disease also in his 50s. So for this reason, Bob, along with his doctor, decide to get genetic testing. Next slide. So in Bob's case, he learns that he is positive for a GBA mutation, one of the more common GBA mutations called N370S. This is a risk factor mutation. So I mentioned this earlier, but GBA mutations have about a 5 times increased risk of Parkinson's disease. But remember that carriers of GBA mutations, the majority of them, will still not develop Parkinson's. And siblings or children will have a 50% chance of sharing that GBA variant. But because of that very incomplete penetrance, sometimes it can be hard to really wrap your mind around what that risk means for somebody. And the other things that I mentioned here is that GBA Parkinson's has a modest increased risk of cognitive impairment, as well as hallucinations and more rapid disease progression. And we talked about that. I showed you some of the data on that. And patients with GBA mutations are now potentially eligible for clinical trials looking at experimental drugs that increase the activity of this particular enzyme. So knowing that you carry this mutation may allow you to enroll in one of those trials. And before we know it, if one of those trials proves successful, having this mutation may allow doctors to prescribe this medicine if the drugs are approved. Next slide. Okay, my second to last case involves Steve. He's 66-year-old. He's had Parkinson's disease for 8 years. He frequently acts out his dreams during sleep. Many of you may be familiar with that symptom. But he has no known family history, but still worries about the potential risk to his children and grandchildren. So he decides to undergo genetic testing. Next slide. So in Steve's case, the genetic test is largely negative for Parkinson's disease genes. There's no high penetrance gene discovered. That's a causal mutation. He doesn't have any of the risk factors that are the lower penetrance risk factors. However, in Steve's case, I'm giving you an example of a more comprehensive genetic test, which I referred to, in which there was an incidental discovery of a mutation in another gene that has nothing to do with Parkinson's disease. And so one example of such an incidental discovery would be the gene BRCA1, which maybe you heard of. It's sometimes called BRCA1. A mutation in this gene leads to an 80% increased risk of breast cancer in female carriers, 50% risk of ovarian cancer, and half of all children are expected to inherit and carry this cancer-causing mutation. So I just want to mention this as an important consideration, and this is why genetic counseling is important if you're thinking about gene testing for Parkinson's. When you have comprehensive genetic testing of the sequencing type, you're often given a choice of receiving these types of incidental findings. They're rare. We estimate about 5% of the time an incidental finding will come up like this. But when they do occur, obviously, they're really important to know about because finding a mutation like this actually changes recommendations for best clinical practice. Individuals who carry BRCA1 mutation usually have surveillance for cancer because they're very high increased risk. So the important kind of take-home message here is that in thinking about genetic testing, we also have to think carefully about potential incidental, medically actionable findings that might come up. And it's not just you that might be affected, but obviously these types of findings have serious implications for your family. In fact, usually when a mutation in BRCA1 is discovered, we are advising patients to tell all of their family about this potential risk because it really is something that needs to be followed if it exists. Next slide. So our last case is Alice. She's 65 years old and she's diagnosed with Parkinson's after noticing that her handwriting is kind of getting smaller. She also notes that she's no longer swinging her right arm when she walks. And her grandfather also had a tremor in his 70s, but wasn't ever diagnosed with Parkinson's disease. Next slide. So she has genetic testing because she's curious. And in Alice's case, no high penetrance Parkinson's disease causal mutations are identified. So nothing like LRRK2 or GBA or alpha-synuclein. However, Alice also had a comprehensive genetic test that looks at some of these more modest risk factors. And I'm giving you two examples of such risk factors. These are gene mutations that have very small increases of risk, like 1.3 times or 1.1 times the population risk. This is really a very tiny increased risk, but these are the types of risk factors that we think are actually important for Parkinson's disease in most patients with Parkinson's. And so actually Alice's results represent the most common results when we send genetic testing for Parkinson's disease in patients. And that is we don't discover a high penetrance gene cause, but we will only discover these types of modest risk factors. And we also have to keep in mind that at the moment, our genetic knowledge in Parkinson's disease is incomplete. So over time, we may discover genes that were not included in the test that Alice obtained. Also, of course, there are non-genetic factors that likely play a role, which we're still trying to understand. Next slide. So this is my final slide, and I just wanted to engage you, and perhaps you can give me some feedback during the question and answer. What would you do? Have you already had genetic testing? Would you be interested in genetic testing if you were in the situation that many of my case patients were in? And I'm just quoting 3 different studies that asked patients with Parkinson's disease about their interest in genetic testing. And what we've documented, and we've seen this also at Baylor College of Medicine, is that the majority of our patients are strongly interested in learning genetic risk. They're interested in understanding genetic test results, even if it wouldn't change the management of their Parkinson's disease. Although I think we're rapidly heading towards a place where these types of testing results could actually help us with management or prognostication. And with that, I just wanted to thank you all for your attention. I know this is a complicated and dense topic, but I hope there are some pearls that you're able to take from this. And I'm looking forward to hearing your questions in the question and answer period.
Okay, thank you, Dr. Shulman. We really appreciate all of that information. This has been a lot of great stuff and exciting research really to think about. We're going to take a brief break while we kind of get set up for question and answers. So there's still time to submit your questions. Go ahead and put those in the Q&A feature at the bottom of your screen now. We're going to take a little brief commercial break, maybe about 3 minutes, and then we'll come back with the questions that you all have given us. Thanks. All right, thank you for bearing with us while we wrapped up all these questions that you all have submitted. So I've got my laptop here, so I'm going to be kind of shifting my gaze down so that I can read the questions that you all have submitted throughout our break. Dr. Shulman will go ahead and unmute you so that you'll be able to provide some answers for us. Lots of good questions, actually. Lots of thank you very much. This was very complete information. They appreciated hearing about it. Lots of people who are interested in participating in genetic testing, or maybe who've done some of it already. So I'm just going to jump in and get started. There's not a whole lot of overlap, so there are lots of just totally separate questions. So I'm going to start with this one, which is, have researchers ever come across a family with multiple diagnoses of Parkinson's and not been able to find a genetic link?
Absolutely. There are many families like that where we have multiple affected family members. All Parkinson's neurologists that are geneticists like me have a collection of families like that. When we see patients who have strong family history, we will obtain blood samples. We'll take careful history and draw those pedigrees. In many cases, we will, when we study those samples, we'll identify known gene mutations, but in a minority of them, we won't discover changes in the genes that we might expect. And we believe that those families have new genes, right? Novel genes as causes of Parkinson's disease. I mean, I think if Parkinson's is recurring in families, we almost certainly believe that it is genetic. But we know that there are many genes out there that we have yet to discover. And that's why, as geneticists and scientists that research the genetics of Parkinson's, we try to collect these families for our research projects because it leads us to new genes that we think could help us better understand the disease and hopefully develop new therapies.
Right. Okay, thank you. So we got this question, says, my wife recently died after living with Parkinson's for 29 years, and her nephew also died that same month after living several years with ALS. As both conditions are neurological in nature, is there any connection between them? And if so, could there be some distorted gene that leaves other members of that side of the family susceptible to neurologic abnormalities in their future? And then there is kind of a second part of that, but I'll let you answer the first part first.
Sure. Well, and first of all, I'm really sorry to hear about your wife and your nephew. I mean, that's really difficult to hear. I mean, hearing a brief snippet like that, it's hard to say. I mean, unfortunately, families can be unlucky and you can have two diseases occurring in the same family and they can be unconnected. At the same time, physicians have a strong tradition of applying Occam's razor and trying to connect things. Neurodegenerative diseases like Parkinson's disease and ALS do share certain aspects of their physiology and pathogenesis, meaning that there could be genetic links. It's hard to say without knowing more. Also, it'd be important to understand the specific relationship of your wife and your nephew. But there's one gene that I can think of offhand that can cause a Parkinson's-like syndrome and also can cause an ALS-like syndrome. But there aren't many. Off the top of my head, I should say, because I have to think about it very carefully and systematically. There's not a simple gene mutation that causes both Parkinson's disease and ALS. There are some gene mutations that people have proposed that might have that type of relationship. But they're yet to be proven in most cases.
Okay, thank you. The second part to that is, are there any tests that would indicate susceptibility to neurological abnormalities, or are there any investigations for tests that would help neurologists to eventually find a root cause for these types of diseases? He's got a daughter that would be very interested to help if that were the case.
Right. So, there's a lot of research on trying to identify Parkinson's early in people who don't have the classical syndrome and can be obviously diagnosed by a neurologist with Parkinson's disease. And occasionally, I will have patients who come into my clinic and they tell me that they have a strong family history of Parkinson's disease, and they're just simply interested in getting a checkup to see if they might have early disease. They want to understand their risk. But I often tell them that, in terms of actual clinical tests that we know work, we don't have clear evidence for how to do that right now. It's an active area of research to develop better testing tools, which we call biomarkers for very early disease. But probably the best thing that could be offered to, I think, your daughter, which is who you were asking about, would be that type of visit with a neurologist, or short of that would be a genetic test to look at genetic causes of Parkinson's or ALS. But the likelihood of finding those type of results are probably quite low. And remember, those would be out-of-pocket costs. They could be very expensive. So one would have to consider carefully whether it would be appropriate and worth your while and money.
Okay, thank you. This question comes from Michael, who said he participated in a study and was told that he had a Parkinson's mutation that is recessive. Does that mean that, I think he's asking, does that mean that my child has a 25% chance of getting Parkinson's? And is that only true if, or is that only true if my wife has a recessive Parkinson's gene or something else?
Yeah, so that's right. So it sounds like, you're a budding geneticist, because you understood correctly that if we understand the question correctly, if you have a recessive cause of Parkinson's disease, then you really can only pass along the carrier state to your children, and it would require that your partner, the mother of your children, would have to be a carrier as well, in order for one of your children to develop Parkinson's disease, if you have autosomal recessive Parkinson's. So typically that's unlikely, because most of those autosomal recessive forms of Parkinson's disease, the mutations are pretty rare in the population. It's not impossible, but more likely than not, your partner would not be a carrier. But there are tests available. This is exactly the kind of situation that genetic counselors are usually consulted for, because they can talk with you, they can describe the risk, they can advise on what the appropriate test would be. So that's what I would advise you if you're interested in pursuing it further.
Okay, thank you. This next question says, you said at the start of the presentation that you think Parkinson's is influenced genetically very widely, but that's different than what I've heard, and that it's really only about 10 to 15% of Parkinson's cases are influenced by genetics. Is this a new school of thought?
Yeah, it is. And I hopefully have, converted most of you to the new school of thought. But I think when people were talking about the 15%, they were really thinking about single gene causes of Parkinson's disease, and much more simple genetic inheritance models. It's true that most Parkinson's is not familial, and what we typically refer to as familial Parkinson's disease is like a first-degree relative with Parkinson's disease, meaning you got it directly from your father, and that it's also present in your siblings. Those remain at about 15%, but I think what we've learned in that is not synonymous with genetic Parkinson's disease. That is something, it's important, we should notice it, but most Parkinson's, if not all Parkinson's, is actually influenced by genetics. It's just not single gene, one gene causing disease, similar to the way we used to think about it. So that is a new school of thought, and that's what's changed.
Perfect. Thank you for answering that one. All right. Is the science of genetics sufficiently advanced and reliable to conduct genetic testing of embryos? And if so, what are these companies?
It's a great question. So, this is an area that's at the fringes of my expertise. So I just want to point out that caveat. But there is, there have been many advances in prenatal genetic diagnosis, which is kind of what you're referring to. I think the prenatal genetic diagnosis is not something that we typically think about for Parkinson's disease, because Parkinson's disease is typically not something that people are thinking about at the time of pregnancy, considering that our patient population with Parkinson's disease is typically in their 50s, 60s, 70s or later. So we don't. I've never been involved in a case where we're considering prenatal genetic testing for Parkinson's, and arguably most genetic counselors would not advise that should be considered. But one interesting point, and I kind of put this out there as an asterisk. This is a question that I'm mostly kind of going to skirt around, because I think we would need to get input from more experts from like, obstetrician, gynecologists, and genetic counselors surrounding this. But one interesting point is that the GBA, glucose reversidase gene, which is a really important gene for Parkinson's disease, is actually a cause of another disease as well, which you might have heard of, called Gaucher's disease. And Gaucher's disease is a recessive disease, and so carriers of GBA mutations can develop Parkinson's disease, but those who have 2 copies of the GBA mutation develop Gaucher's disease. And the reason I bring it up as an example here is that separate from testing embryos for gene mutations, like your question is asking about, most pregnant women, as part of routine prenatal care, are offered prenatal genetic testing. And on most prenatal genetic testing panels, actually, frequently they include testing for Gaucher's and some similar disorders. So we are beginning to encounter patients who are young, childbearing age kind of women or men who will come to us having discovered that they are carriers of GBA mutations based on prenatal genetic testing and asking us about risk of Parkinson's disease. And so this is a new world of the neurology clinic where some of these people are learning about risk of Parkinson's, prenatally.
Okay, so I'm going to go back, kind of loop back to a couple questions that we've received, kind of back to this familial Parkinson's idea, which is, so one question was, I have a family history of Parkinson's, but don't have the diagnosis myself. I want to know my risk for developing PD. When should I pursue genetic counseling? How do you ask your primary care doctor to help with this? And then also kind of lumping in another question that came in, which is, how worried should my kids or grandkids be? This is a separate question. Someone who does have Parkinson's diagnosis, how worried should my kids or grandkids be?
Right. So, I mean, regarding the first situation, I think you can talk to your regular doctor about, whether they would advise genetic testing. Hopefully you got a sense that in most cases, people who have some modest family history of Parkinson's disease, genetic testing may well be negative. And so I wanted just to kind of prepare everybody for that. Even those of you who have Parkinson's disease, the majority of our testing that we do, turns out negative. And so, we don't always find the cause and we don't know all the causes. So, genetic testing may become better over time. And also, applicable to the first question, again, it's likely that, testing would not be covered by insurance. And even the conversation with a genetic counselor is not likely going to be covered by insurance. So, the truth is, and, I think it's a sad truth, my personal opinion, is that, to go on that journey of understanding your risk and your family's risk is probably going to be associated with some out-of-pocket costs. To have the appropriate genetic counseling and get testing. Now, it could still be worthwhile, but you have to be aware that those costs exist. And I think much of what I said would also apply to the second question. For that second questioner, it sounded like that was a situation where somebody knows that they have Parkinson's disease already. They're interested about genetic risk for their children. What I would advise that individual is to look at the handouts. I think that would be somebody who would be a perfect candidate for the PD generation study, which it offers free testing of the 7 most important genes that we test for in Parkinson's disease, and also offers genetic counseling, and that's all covered. The alternative, if you don't want to enroll in the PD generation study, would be to bring to the attention of your neurologist the InVitae sponsored gene testing program that I mentioned, because that is a genetic testing program that is free, and they offer a good, fairly comprehensive genetic panel that could be applied. Unfortunately, I don't think either PD Generations or InVitae offer those programs to people who have a family history of Parkinson's disease, but don't actually have Parkinson's disease themselves. So they wouldn't apply, I don't think, for the first question.
Okay. Thank you. Tom would like to know, will genetic engineering in the future be able to alter the structure of Parkinson's disease?
Right. So, there, in thinking about the way genetics can lead to new therapies, one way is to take a target that relates to a Parkinson's gene like alpha-synuclein and develop something that attacks that protein, right? Something like an antibody, which I mentioned, or you could imagine a small molecule drug that you take or you're injected with, and then attacks the protein. Genetic engineering typically refers to approaches to actually manipulate the genome directly. This can sometimes be done with viruses, for example. And that type of genetic engineering therapy there's interest in it for Parkinson's disease. You could imagine making changes deep within the brain to the regions that are vulnerable in Parkinson's that might affect alpha-synuclein or other biological pathways. I wouldn't say that any such therapies are going to happen, next month. I mean, they're kind of a long road to develop a therapy like that and to get it approved. You can imagine there's a lot of questions about safety. There are, short of that, there are some interesting, exciting, potentially promising approaches to use gene therapy-like approaches to reduce the alpha-synuclein protein levels in people that might be at risk or have Parkinson's disease. And so, that might be the closest thing. In certain animal models of Parkinson's disease, the last thing I'll say on this, like mice and even primate or monkey models of Parkinson's, people have tried experimentally using gene therapy-like approaches to treat Parkinson's disease. So it's not science fiction.
Good to know. All right. How do atypical forms of PD fit in genetically? Are they different? How do those kind of show up in test?
Well, there are two ways of answering that question. I think probably what the questioner is referring to when they say atypical forms of Parkinson's might be things like dementia with Lewy bodies, progressive supranuclear palsy. These are some other disorders that we often refer to as kind of atypical Parkinson's because they can look like Parkinson's, but we typically think of them as alternative diagnoses. Those are different diseases, and they have their own genetic panels, their own genetic architecture, so many of what I covered in my talk would be applicable to those diseases, but there are some different genes involved that would have to be considered. But the other way of looking at that question on atypical Parkinson's is to kind of get back to my my idea that I think all Parkinson's is atypical. I don't know what typical Parkinson's is, honestly. I'm being a little joking with this, but what I mean by it is that Parkinson's is really very heterogeneous. I think each patient's Parkinson's is their own unique form of Parkinson's, and I think genetics absolutely is the reason for that. Genetics is the substrate of variation in the human population, and it almost certainly accounts for the unique attributes of Parkinson's. I'll just say, I think there was a question, Kathleen, I have to admit, I scrolled through some of the questions, and someone was asking about why it is that Ashkenazi Jews, for example, have a different penetrance when it comes to the LRRK2 gene mutation. And so I just want to bring that question up in answering this question here, because we really think that the genome and genetic variation, mostly which we still don't understand, modifies even the genes that we know about. And so when we talk about variable penetrance for something like LRRK2 or GBA, this is almost in large part due to other genes in the genome that modify the penetrance or the expression of LRRK2 or GBAPD. And that's of course varies by ethnicity, because the substrate of different ethnicities is genetic makeup. And so that is really what's modifying disease and the effects of certain genes that we know about in different populations.
Okay, great. Well, that was going to be my very next question. So you beat me to that one. Richard would like to know, have there been any studies on factors that affect incomplete penetrance, such as diet, chemical exposure, lifestyle?
Right, that's a great question. There is certainly a lot of research now on lifestyle interventions in Parkinson's, like exercise and diet. That's a very hot area. I can't say for certain. I'm sure some of those studies are also, doing genetic testing, and so they'll be in a strong position to understand how lifestyle interventions modify genetic factors and interact with genetic factors. As I mentioned, we also think of, in some cases, in most cases, there's going to be a lot of gene-gene interactions, right? So genetic factors can interact with one another. Genes will modify the penetrance of things like LRRK2 and GBA. But I think we have to think about lifestyle factors. If somebody is a GBA carrier and they're worried about their risk of Parkinson's disease, I would encourage them to exercise. We may not know that exercise can be preventative for Parkinson's, but we know that exercise affects many other medical, is an important way of reducing other medical risk, like cardiovascular risk. And maybe there's a chance that it alters the penetrance of mutations like GBA.
Okay, thank you. Janet would like to know, once tested, do you stay in a database for future evaluation as science continues to evolve and understanding changes, so that they can go back and use your information for future research?
It's a great question. It really depends on the specific genetic test, who does it. Not necessarily with most commercial tests. There are certain research projects that are doing testing where they will, maintain information and have an opportunity to review it as new knowledge is gained. I mean, this is a really great question. It's something that the field is struggling a lot with. It really applies to the kind of comprehensive whole genome or whole exome sequencing tests that I referred to. Those tests gather a lot more information than what we currently know about the genetics of Parkinson's. And so what typically happens is that we gather the information, and then the data has to be analyzed using current knowledge, and a report is generated. And so then, what this questioner is asking is, 2 years later, can a new report be generated, right? And how do you develop a system to do that, if ideally automatically, to incorporate new knowledge? What should the cost of that new test, newly interpreted test be, even if it doesn't require new blood or new analysis? This is an area that the field is really trying to work out now. I will mention, like, for example, Baylor Genetics, which is a private-public partnership that the Baylor College of Medicine is involved in, does offer an exome sequencing test where there is an option at some time later to pay for a reinterpretation. And I think other sequencing tests offer that type of option. It's essential because knowledge is increasing literally, month by month. So you do want a capability after some time to be able to relook at the information and create a new, improved report.
Okay. Blair had a great question. I think it kind of encapsulates a lot of what some of people's concerns are with genetic testing, which is, if I have significant genetic testing results, how do insurance companies react to this information? Does it affect my ability to obtain affordable insurance? Is there government policy regarding this concern? Can you comment on that?
It's a great question. It's an important question. Genetic counselors are really the experts when it comes to answering questions like that. And this is another reason why, I advise to seriously consider talking with a genetic counselor before you obtain genetic testing. Usually when you obtain genetic testing, even if you don't speak to a genetic counselor, some information will be provided about this. There are some important laws that protect against genetic discrimination. But honestly, like, in general with all laws, they're complicated. Based on my best current understanding, the laws may not be operable if you're already known to have Parkinson's disease. When people have known conditions already, I think I believe, and again, I have to apologize because I don't know all the details, but I believe that you're not protected in terms of the specifics of the genetic tests and possibly being used. But I think the important point that, is brought out by the question is that, genetic information potentially has implications for, insurance. It's important to know those risks before you proceed. The same is true, of course, of just any other aspect of your medical history, right? I mean, the fact that you might have a diagnosis of Parkinson's disease could be used by insurance companies against you. So there are some important, unusual, and specific things that apply to genetic testing, particularly those who don't have a disease, right, but might be at risk in the future because of that genetic test. But I think also this is my personal belief, is that we sometimes imbue a certain magical quality to genetic testing. But I don't think it's that different from all kinds of tests that we do, blood tests, brain scans, other aspects of our medical record. I think most of the types of risks and benefits are kind of similar. I imagine years ago when MRIs started to be used, and people were like, coming home with these CDs with MRIs, everyone was like freaking out, perhaps briefly, that we can't give patients their MRI CDs to look at. It's going to cause some kind of chaos. But I think obviously, if any of you have had a brain scan, you can leave the facility with a little CD that has a copy of the scan, and the world hasn't fallen apart. I really think genetics is moving in that direction, where we're all going to basically get a CD with our comprehensive results, and it'll stimulate and conversation with your healthcare team about what its implications are.
Yeah, that's a good point. Okay, I want to be mindful of time. We do have a couple of pretty additional interesting questions that I want to throw out there. Some of these are some, genetic relationships to Parkinson's and other disorders. So one is, is there a genetic relationship to celiac disease and Parkinson's?
No, I don't know of anything definitive off the top of my head. So I'd have to look it up.
Okay. The other is from a woman, Cheryl, who says that wants to know if there's a genetic connection between essential tremor and Parkinson's. She had a mother, brother, sister have essential tremors, and she has both.
That's a great question. We've actually done some research on this topic, but I can say that there's no definitive genetic connection between essential tremor and Parkinson's disease. There's a lot of debate within the neurological community about what, if any, relationship exists between essential tremor and Parkinson's disease. And the possible genetic link would be an even more advanced version of that debate and controversy. We did some research to try to look at this. We didn't find anything, but I think it's something that needs to be studied in greater depth. Okay,
thank you. So you talked about getting these results, and I'm just kind of paraphrasing from a number of questions that we got about getting, if you choose to opt into genetic testing, can any neurologist help you interpret these clinical genetic tests? Or is that something that just people have kind of limited specialty in? How does that work if people were to pursue that? Should they also be seeing a geneticist, or can the neurologist just kind of manage all that?
Yeah, well, these are great questions. It's really going to come down to the comfort level of the individual neurologist. The field is rapidly changing. That's one of the things that drew me into medicine, right? Is that, you do have to keep up with things. Things change over time as new knowledge is gained. I think most neurologists and physicians feel the same way, that they, want to be up to date, and they want to be able to, speak intelligently about the latest tests. But, keep in mind that, the genetics of Parkinson's disease, is a rather new field. So many neurologists will not have been trained to think about genetic testing in Parkinson's disease. And, there is some apprehension amongst, neurologists about whether they feel confident and capable to advise patients and help them interpret their tests. One of the things that the PD generation study is doing is actually comparing, how patients feel. So this is the study that I mentioned from the Parkinson's Foundation. One of the things they're studying is how good neurologists do in counseling their patients compared to genetic counselors. So they're doing a little bit of a head-to-head comparison, and they're talking to patients afterwards and saying, did you feel like things were explained well to you? I mean, like anything, basically the answer to your question is we need to study this. We need to understand how to provide the most effective care and explain complicated test results to patients. It's complicated on many levels. One complication that we're referring to is practitioner neurologist apprehension, knowledge that maybe they never received in their training and learning to take care of patients with Parkinson's. But another is just how to explain some of these concepts. I just spent an hour or more with most of you, and kind of gave you a pretty high level overview of some of these genetic issues. Something like the challenge of telling somebody if they have a GBA mutation that they have a 5 times increased risk, but that means that 90% of the time they're still not going to get Parkinson's disease. Unpacking that takes time, and as most of you know, most clinical encounters when you see your doctor are somewhat time limited and rushed. So there are doctors who worry that just even fitting in the time to fully discuss these tests is challenging. So this is why genetic counselors are sought after often.
Okay, well, like I said, I want to be mindful of time. I know that we still have questions in the Q&A. I'm sorry that I couldn't get to all of them, but if you have questions that you are just really wanting answered, you are welcome. I don't know, Dr. Shulman, if you can stay on just a few more minutes afterwards, and maybe if there are brief responses that you can give to type in some of those answers as you go through them on your end. But otherwise, if you want to email me, my email address is Christ@HAPSonline. That's CRIST@HAPSonline.org. And you're more than welcome to email me those questions that we didn't get to. And I will be happy to try to get Dr. Shulman to get us some answers so that we can get those addressed for you. You all did such a wonderful job of submitting a whole lot of questions. Several of you did ask about getting the handouts. Again, you can go to hapsonline.org, and under the summer lecture series page, you can find the handouts for today, the full packet, the full program that also has the slides for Dr. Shulman's presentation. And there was one final question that will be easy to answer. I assume that you have a clinical practice in the Houston area. Are you accepting new patients? I'll let you just give a yes or no on that one.
Yes, I am at Baylor College of Medicine. And I'll just add, probably the easiest thing as far as the residual questions, Kathleen, if you're able to just send me a transcript of the questions and answers, if that's possible, I can create answers to them, and then they could be uploaded on HAPS or sent to the individual questioners. I'm happy to do that. I think that's a lot easier than me, responding to each individual one on my phone. So
Yeah. Oh, sure. Yeah, right. I keep forgetting that you're logged in through your phone. So no problem. Okay. So we will figure that out. And then we'll try to figure out a way that we can upload the answers to those questions and get those on our website. So that I'm going to go ahead and tie a bow on this question and answer session. Again, thank you to all of you who submitted such great questions. Next up is going to be the Movement Lab with Jeffrey Wu. And if you're not able to stick around and move with us, then I want to take a minute to thank you all for joining us and to again thank our sponsors for their generous support of our educational programs. This session will be available online in the coming weeks, so don't forget to check out our website for the latest on our in-person and online exercise sessions and other educational programs coming up, including the symposium that we'll be having later later on in September, which is scheduled Saturday, September 18th. So now give us a brief minute. We're going to jump over to Jeffrey Wu, who's going to lead us in full body movement activity. Jeffrey is a physical therapist at Tier Memorial Hermann Outpatient, where he evaluates and treats patients with neurodegenerative diseases. He received his Doctor of Physical Therapy degree from Nova Southeastern University in South Florida and is LSVT BIG certified. Beyond working with the Parkinson's population, Jeffrey's interests include treating spinal cord injury and amputees. He enjoys mentoring and serving as a clinical instructor to physical therapy students, and he is a very popular HAPS instructor and teaches the full body aerobics class on Thursday afternoons on Zoom. So if you will hold on for just a second, we're going to jump over to his session with him right now. Thank you again.
Hey guys, I'm Jeffrey, and today I'll be going over the Movement Lab as part of the summer lecture series. And we will be going over exercises in both sitting and standing. We'll also be using a prop, a small ball, and if you can't find one, you can also use a toilet paper roll. And as always, make sure you do these exercises at your own pace and use hand support as needed for safety. Let's get started. All right, guys, let's start with some warm-up in sitting. So sitting in the chair, we're going to start with our arms reaching down low towards the ground and then reaching those arms high up towards the ceiling. And then breathing out on the way down and a deep breath in on the way up. And try to feel a nice stretch. So reaching down and reaching back up. And we'll do this back and forth. We'll do 3 more. That's 2, and we got 1 more. Nice deep breath, very good. And then our next one is going to be reaching to the side. So we can start with left forearm onto the left thigh. And then with your right arm, we're gonna reach across towards the left side. And then we're gonna switch. So right forearm on the right thigh and left arm, you're going to reach to the side and try to feel the stretch all the way on the side. And we'll do this back and forth. So a big reach to the side. Alternating. Back and forth. And we'll do 5 more on each side. 1, 2, 3, big reach, 4, and 1 more, 5. All right, now we're going to warm up our shoulders. So with the first one, you're going to have your arms to the side, but not completely against the side of your body. So a little space here. We're going to start like this, and with your hands open, we're just going to rotate your hand out, just like this. We're going to come in and open back out, back and forth to warm up the shoulder joints. You can feel a little stretch in the front of the shoulder. We'll go like this, back and forth. The movement does not have to be fast. Keep it nice and controlled. And we'll do 10 of these. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Very good. Now our next one, we're gonna bring our shoulders up to the 90-90 position, shoulder level, and we're gonna do the same thing. We're gonna rotate our shoulder out and back down 10 times, up and down. 1, 2, 3, 4. Good. 5, 6, 7, 8, 9, and 10. All right, very good. Our last warm-up is going to be arms nice and wide to the side, and we're going to do the twist. So we're turning our body to one side, twist, and try to clap, open back up, and turn to the other side, and open back up. So I want to see a good rotation with our upper body. Turn and open. Good. And we'll go back and forth, and we'll also do this 10 times. 1, 2, 3, 4, 5, 6, 7, 8, 9, one more, 10. All right, with our next exercises, we're gonna do them using the ball. So grab your small ball, and we're gonna start by warming up with the ball. So sit with your knees close together, and we're gonna start by bringing the ball under both legs like this. So we'll go one direction. So right now I'm going counterclockwise. So just like this, as a warm up, keep it going. And let's go a little faster. Yep, keep going. All right, very good. Now we're gonna go the other direction, so clockwise. All right, let's speed it up a little faster. All right, very good. Now we're gonna try to go between the legs. So with the ball, you're gonna start from inside. It's gonna come out under the right leg. You're gonna bring it up with the right arm and then bring it down the middle under the left leg and then back out with the left arm back to the middle underneath the right. And then with the right arm, you're gonna bring it down to the middle underneath the left. So let's do this back and forth. So switching the ball between each hand. Yep, just like this, like you're dribbling in between the legs. Good. Yep, keep the movement going, and then we'll try a little bit faster. Good, very good. Keep going, a little faster. All right, very good. Now, our next one, we're still going to use the ball, and this one is going to be a step to the side. So we're going to start with sitting up nice and tall in the chair, and then with the ball, we're going to bring our right leg out, and at the same time, bring your right arm out holding the ball. So one more time. So to the right, we're going to step out like that and come back to the middle. And then we're going to go to the other side. So to the left, bringing that left leg straight out and left arm out while holding the ball. And we come back to the middle, and we're going to switch hands to the other side with the ball and come back and then out to the left. So let's do this back and forth. Out. And to the left and to the right. We'll do 10 of these. 1, 2, nice and big movements. 3, 4, 5, 6, 7, good. 8, 9, and 10. All right, very nice. All right, the next one is going to be the twist. So we did the twist before, but that was without the ball. This time we're going to use the ball. So we're going to start with our arms out to the side. And when you turn and twist, you're going to grab the ball and bring it out to the other side and turn, grab the ball, bring it back out to the right side. So I turn to the right, my left hand's going to grab the ball, bring it out to the left side, and just like that, back and forth. And we'll also do 10 of these. So starting now, 1, 2, 3, 4, 5, 6, good. 7, 8, 9, and 10. All right, very good. Now our next one is we're going to move the ball in the diagonal position, and we're going to hold on with the ball with both hands. So we're going to start down low. So for me, let's start with the left lower side, and we're going to bring the ball up to the right upper corner. So we're going to start left lower to right upper, and we're going to use this chop motion. So up and down, up and down. Yep, we'll keep it in this direction for 10 times. So 1, 2, 3, good. 4, 5, 6, 7, 8, 9, and 10. All right, very good. Now we're going to go the other direction. So now it's the right lower and left upper. So up and across, down and up and down. We'll go 1, 2, 3, 4, 5, good. 6, 7, 8, 9, and 10. Awesome. All right, we're going to get ready for our standing exercises. All right, guys, we're going to start with some sit-to-stands, the big sit-to-stands. So to do this, you're going to start by leaning forward with your hands reaching straight out, and we're going to stand up big with your arms coming out to the side. And we're going to reach back forward again as we slowly sit back down. So reach forward, open big, reach forward, and down. So we'll do 10 of these. Nice and big sit-to-stands. Starting now. Ready, and go. 1, 2, 3, 4. Keep those arms big. 5, 6, 7, 8, 9, and 10. All right, very good. Now, the same thing, this time we're going to add in the use of the ball. So when you lean forward and stand, the ball is going to be in one hand as you open your arms out. And when you come down and stand again, you're going to switch the ball to the other hand. So it looks like this. Come forward, ball's in my right hand, lean forward, sit, stand again, the ball's in my left hand. So we're going to alternate and switch each time. And we're going to do 10 times. Ready? And go. 1, 2. Make sure you're leaning forward still. 3, 4, 5, 6, 7, 8, 9, and 10. All right, very good. Now, we can move the chair aside, and you can use it to hold on for support if you need it for balance. But our next one, we're going to do it in standing. We're going to do the big march. So when we march our right knee up, we're going to bring our left arm up. So the opposite arm, opposite knee. Left knee up, and the right arm will go up. And then we're going to switch. So switch the ball to the other hand, march, and march. So right knee up, left arm up, switch, left knee up, and right arm up. So we're going to do this back and forth. Big march. Up and switch. Try to make sure you're switching the ball in each hand. And we'll do 10 marches. So 1, 2, 3, 4. Good. 5, 6, 7, 8, 9, and 10. All right. And for the next one, we're going to do a side-to-side reach. And we're still using the ball. If you need to use a chair, you can have the chair in front of you. And we're going to start reaching to the left side. We're going to shift our weight to that left side. And with the left arm, you're going to reach up to the side. And we're going to switch to the right side, shift to the right, and reach your arm out to the right. And we're going to switch the ball back to the left side. As you reach as high as you can, really try to stretch and keep your body in that straight, long line. And reach to the other side. And if you can, try to tippy toe on the other leg, but make sure you have the right balance for this. And reach back to the right. So we'll go side to side reach, left and right. And we'll also do this 10 times. So 1, and switch. 2, 3, big reach. 4, 5, good. 6, 7, 8, 9, and 10. All right, very good. Now our next one is going to be a side step, a big side step, while holding on to the ball. So make sure you have enough space to the side. You're going to start in the middle, holding the ball with both hands. And when you step out to the right, a big step out to the right, and you're going to open your arms out big, and the ball will be in the right hand. And then bring it back forward, and then step out to the other side, the left. Big step to the left, arms out, ball into the left hand. Come back to the middle. To the right, and to the left. And we'll do this back and forth. 1, 2, 3, Keep the movements nice and big. Good. 4, 5, 6, 7. Keep going. Good. 8, 9, and 10. All right, very good. Now our next one is we're going to do the twist. So we did that in sitting before. Now we're just going to do it in standing. And you're going to start with the feet about shoulder width apart. So you're nice and stable and arms out to the side. So we'll twist and open and twist to the left and open back up and switching the ball in between. And in standing, you're allowed to twist with your hips and your leg a little bit more as you turn and rotate to that one side. So we'll practice more with that. So turn and open, twist to the left and open back up. And we'll do 10 of these. So starting now, 1, 2, 3, 4, 5, 6, 7, 8. Good. 9 and 10. All right, very good. Now our last one, we don't need the ball anymore. We're gonna work on some stepping and also shifting our weight forward and back. So I'm gonna show you guys from the side view. So when we step we'll step with the right leg first. So when the right leg steps forward, we're going to shift our weight forward onto that right leg and the left arm comes up. So we're gonna step forward and then we're gonna step back. So when you step back, the right arm will come up. So again, step forward with the right foot, shift forward, left arm comes up, come back to the middle, and then we're gonna step back with the right foot and the right arm swings forward. So it looks like this. Stepping forward and stepping back. We're moving just with the right foot. So right step forward and right step back. So let's work on this. 10 on this right side. 1, 2, 3, 4. Good. 5, 6, 7, 8, 9, and one more. 10. All right, very good. And now we're gonna have the left leg, that's the one that's going to move. So with my left leg, I'm gonna step forward and my right arm will go forward as I shift my weight forward, come back to the middle and step back with the left leg and my left arm will come forward. So it's the same thing, now we're just doing it to the opposite side. So that's 1. That's 2. And it's 3. Good. Step forward, shift your weight forward, back to the middle, step back, shift your weight back a little bit, and come back. That's 4. 5. 6. Good. 7. 8. 9. And 10. All right, guys, that will conclude the movement lab for the summer lecture series. Hope you guys enjoyed it. See you guys next time.
Okay, well, thank you, Jeffrey. That is a wrap for both our August session and the 2021 HAPS Summer Lecture Series. We look forward to seeing you back online in September for our virtual biennial Engage, Educate, Empower Symposium. We hope that you'll join us for that full day program where we'll have a lot to offer, including an interactive platform to visit with many of our community partners. Thanks again to Dr. Shulman for all of his wonderful information and expertise, also for supporting our fun summer theme here at the HAPS office. And just to those of you who are at home, as a reminder, HAPS is always here to help. So please reach out if there's anything that you need. Have a great day, fantastic Saturday, and awesome rest of your summer. See you in September.